muchas gracias al comité organizador y pues me han pedido que dé la plática en inglés, así que las diapositivas están en español, pero la plática, o sea, me pidieron que la diera en inglés y pues de todos modos cualquier pregunta. Y pues antes que nada, pues otra vez agradezco al comité organizador por la invitación a participar, ¿no? También. Well, first, I would like to start saying that we all know that plants, medicinal plants, have been very important in the development of new drugs, especially with pharmaceutical application. And among them, we can talk about the morphine, vinblastine, vincristine, and taxol. All of them are drugs that have been widely used in cancer therapy, and and also in pain, for the treatment of pain. And all of these metabolites have been isolated from plants. However, normally when we do a plant study, we normally will use something that is called bio-guided isolation. We know the bio-guided isolation will follow more or less that scheme. We will normally have a natural source, in this case could be a plant, and we will do the extraction to get the extract. That extract we will evaluate the biological activity in order to know if it can have any biological or pharmaceutical application. As Dr. Cepeda previously mentioned, you will do a previous evaluation. Then, once you know is active, then you will start the process of trying to identify the active metabolite. So you will do a fractionation using normally different chromatographic techniques or chromatographic methods. And once you have that, then most of the time you have to evaluate every fraction again to know whether there is an activity or not. You have to do a bioassay, and you will keep doing this many times. It could be a work that could take you for over a year sometime, and eventually you will find the active metabolite. Most of the time you don't find it, because sometimes it happened that there was no only one compound being active, if not there were many of them. So sometimes you can spend a lot of time trying to do this fractionation, trying to search, trying to find the active compound, and probably you won't. So what happened in this case is that most of the time this method has proved to be very successful, because most of the compounds that are actually in use, they were isolated using this bio-guided fractionation. However, most of the time, although very successful, it has some disadvantages. Among them, we can say that normally you will need a big amount of plant material to do the work. Sometimes you will have to collect big amount. I mean, I've seen, for example, people collecting 5 kilograms of plants, and then to do the extraction, you have to use a lot of solvent. You will use a lot of solvents, organic solvents, to get the extract, which will have an impact in terms of the cost, and as well an ecological impact as well, because you are generating a lot of waste, chemical waste material. On top of that, every time you do a fractionation, you will have to do this bioassay again. Every time you do a fractionation, you will use big amount of solvents. Then it will end up again generating a lot of waste material, and it can take quite a long time. And again, few times you will get the active compound. And also, because you need to collect sometimes big amount of plant material, it limits the type of plants that you can study. For example, you won't be able to do that work with plants that have a limited distribution. For example, if they are plants that are, for example, are endemic to a small area, and you won't be able to collect 5 kilograms of that plants, and we're talking about plant material. Sometimes 5 kilograms of plant material, that is impossible. Sometimes you will be allowed to collect only three specimens of the plants, for example, in some countries. So you need to find new alternatives, and it's where metabolomics and chemometrics come into play, because they can play a big role in a new way of doing this work instead of doing the bio-guided isolation. So if you do a quick search, you will see that for metabolomics there is a steady increase in the papers that keeps appearing with the term metabolomics. And when you do that for chemometrics, you will also see that there is a big increase. So slowly, but there has been a steady increase in the papers that cite chemometrics. So let's just give a quick definition of what is that, and then we will talk about some examples of an application of chemometrics. When we talk about metabolomics, we have to take into account that the term was introduced in 1998 to refer to the complete set of all the molecules, the low molecular weight molecule that were synthesized by a cell. But at the same time, those were metabolites that were synthesized, but that they also have an impact or can leave a fingerprint of what happened in the cells, what happened in the process. So that's why metabolomics became very huge, because they decided to start studying. Okay, in this case, we can have if you study DNA, you talk about genomics. If you use RNA, transcriptomics, there is the proteomics, and also the metabolomics falls into this area, where you can study carbohydrates, lipids, nucleic acids, and also amino acids. But at the same time, all of this in a plant, that those are the ones that will provide the chemical, the building molecule to produce the secondary metabolites, that are the ones that most of us studying medicinal plants are interested in. So also the secondary metabolites are part of the metabolome of the plant, and that's why it is also called metabolomics. So it emerged the metabolomics appeared as a helping new tool to find out what is happening. However, although in metabolomics we could know what was happening in the cell, we needed a tool to compare and to analyze the data, because we could have only the metabolome. We would have all the data, all the set of information, but we needed to extract something from there. We needed to know what is happening, and that is when chemometrics appeared, because chemometrics is one of the tools that can be used to analyze that data that metabolomics is providing. You get the data from metabolomics, and then you have to use chemometrics to analyze that. And then, chemometrics is even a term that is older than metabolomics. So this is from 1971. So it's Svante Wold from the University of Sweden. It used it to refer mainly to an interdisciplinary field in chemistry that will use statistics and mathematical models in order to explain and understand information that was given from analytical chemistry. When I say analytical chemistry, I refer to mainly it could be NMR, that will tell us the type of protons that are in molecule, IR, that will tell us the functional groups, UV, chromatography that will tell us the different compounds present, and etc. So how do we use chemometrics now? We know that now plants, they are really specimens with lots of different variables. We are talking that every single metabolite in a plant will be a variable X. So you have a plant, you have around 150 to 200 different chemical components, and every component will be a variable X. So you will form a matrix of variable X. So, and most of the time, that is how they, most of the systems that we work, they are not really univariable or bivariable. If not, they will contain several variables that we have to take into account. So most of the time, and all those systems are very complex. So to get clear this, we will use some examples. I would like to say that chemometrics has been used for quality control of plants to determine which variables are important for the quality control of a herbal product, a herbal medicine. It can be used to differentiate between different plant, populations of plants, to try to find the differences between them, because probably one of them is growing in one area that is humid, and probably in another one is growing in an area that is very dry. It's a dry area, so we can find the differences between them. So as well, we can identify geographical differences between plants. So we can say, oh, this plant that is growing in Puebla has these chemical compounds that are specific to them compared to those growing in Veracruz. And even within Veracruz, we can find different differences between what is happening in the chemical component. So it has a lot of applications, really. So I'm just gonna use an example of how we are using this. For example, this Scutellaria bicalensis is used for the treatment of lung cancer, and we wanted to know which are the components that are involved in the pharmacological properties of this plant. So we decided to build eight different extracts from the same plant material. So from the same plant material, we took portions of the plant material, and each portion was subjected to an extraction with different solvents. Of course, being the same plant material, we can expect to get similar chemical components, but probably just differences between the concentration of them. So when you have that, you do a HPLC analysis. In this case, we have been using mainly HPLC. So we can get this fingerprint, we will use HPLC. You can use any other analytical tool. You can use NMR, you can use infrared. So, and then also for each extract, we tested the biological activity. In this case, it was the cell proliferation activity. So once we knew, well, we have here the fingerprint or the HPLC chromatograms for each, for all of them, and at the same time here we had the biological activity that each extract was presenting. So once we have these two sets of data, this is what will be my matrix of only X variables, because, for example, we can see that one variable can be present in various extracts, but in different concentrations. So, in this case, we use two main tools. One of them is called principal component analysis, and the principal component analysis will only tell me what are the chemical differences. It will tell me which samples they are related, which ones are similar, and which ones are different. So we will form clusters. And then each cluster will tell me. And for example, here I could see that I have 3 clusters of data, and at the same time I can know which variables are influencing that they are close together. For example, I can be able to say these plants are together because they have these variables that make them unique. So that is what makes them a group. And at the same time, you can do that for each cluster. So you can identify which variables are influencing or are saying these are similar, they are together because of this variable. So that is the first part that we know. So we do PCA, or principal component analysis, to know which ones are similar and which ones are not. Most of the time, the samples or the extract that have similar activities will tend to cluster together, because at the end of the day, they will be together because chemically they are similar. And if they are similar chemically, you expect them to be as well similar in biological activity. So most of the time, however, it's important to make notice that PCA is not a correlation tool. So you cannot say that for sure this is happening until you do the next test that is called PLS, which is partial least squares. So until you do that, because it's when you do this partial least square, is when you are going to use a Y variable. In this case, is when you are going to introduce the biological activity. You have a value for the biological activity of each sample. So you can introduce that activity as a Y variable to make a correlation. So now you want to know which compounds are responsible for that activity. So that is why, again, PCA will tell you which ones are similar, but you cannot say that it's not a correlation tool. You have to use PLS in order to get that correlation. It will tell you this biological activity is because of this number of variables. So once, for these plants, once we did the PLS, we could see that the biological activity was mainly due because of these three variables. Those were already identified compounds, and actually we kind of knew that those were the active ingredient. However, we were trying to use in this case, we were trying to see if PLS was going to be able to tell us that, if we can use it to relate to biological activity. And indeed, we were glad that we could see that it was pointing towards the 3 components that were active. So in this case, we use it for a plant that we already knew which ones were the biological the active component, the responsible for the activity. So we wanted to know. Now we know that it will show us the active components, and we decided to move and then try to do the same model with plants where we didn't know the active components. So one of the works we did, and we did in collaboration with Dr. Garcia Rodriguez, that will give a talk later on. So we studied the biological activity of different salvia species, and also I saw Dr. Chazaro somewhere around there, and he was also part of this work. Oh, yeah, I can see now. And then we collected this number of plants of salvia, yeah, However, something that was quite shocking is that the number of species for salvia plant that were between 1972 and 1999 was around 27 species, and we only found probably like 13. So that also tells us about how many probably species of plant had been probably moved apart from their natural habitat as well because of the development of the city. So for this, we tried to do and try to find out whether we can, something quite unique here is that most of salvia species are used for inflammatory, for inflammation. Most of them are used for inflammation. However, so far there are no chemical components there reported as the main ingredient. However, all salvias, most of them, well not all, but most of them are used for the treatment of inflammation. So when we did that, we can see that all the peaks that you see pointing downward are ones that are contributing to the biological activity. What we found is that there were various compounds, so that's why probably you don't find a main chemical ingredient in salvia that you can say this is the responsible for the anti-inflammatory effect, and that you can say is very potent, because we can see that there are various that contribute, although there is one that contributes the most. There is one, and so one of the next steps is trying to isolate those compounds, because if you do this you can do it with very small amounts of sample, and then you can know what are the chemical components in the plants that are responsible for the biological activity, and then you can isolate them straight away. So in a more direct form, rather than doing the bio-guided isolation that will take you forever, or could take you forever, really. We did also this for some plants that were used in Anglo-Saxon England, that were used during the Anglo-Saxon period as antibacterial. So for these plants, we could also we found, in this case, the chemical component. Just recently, a PhD student has finished identifying the chemical compound responsible for the activity. But what is quite interesting in here is that for this study, we only used 20 grams of plant material, because we were allowed to collect only 3 specimens of the plant. We weren't allowed to take more than that. And then, for example, if we wouldn't have this tool, we wouldn't be able we wouldn't have carried out this type of work, because we couldn't, we would never had enough material. Recently, we have been studying as well Bidens spilosa, that has been associated to the use for tuberculosis. I mean, we know that Veracruz is one of the states in Mexico with one of the highest incidents of tuberculosis, and at the same time, the death associated to that is also very high in the state. So we decided to do that. Just now, this year, a PhD student joined the group and will try to identify the compound. She managed to identify to pinpoint two main components, really. Two main components that it seems that they are the responsible for the activity of against Mycobacterium of Bidens spilosa. So this one, we will keep working on that. And we have done this work as well for using IR. So it uses infrared for Plantago australis. In this case, it was for cell proliferation to study the inhibition of cell proliferation, especially with tumoral cells, and then in this case we were able to relate the activity to specific parts of the molecule, or mainly functional groups in the molecule. So in these plants, we were able to relate that to two main components that were already known, not in Plantago australis, but in another Plantago. So they were, so we managed to well, in this case, so far, we managed to relate that. However, we haven't continued the work with Plantago. And the last example that I want to talk is this plant that has been used in Yucatan. This was made in collaboration with people from Yucatan. And this plant has been used for the treatment of leishmania. And then we wanted to know, there was a previous study, a full PhD work, trying to do the bio-guided isolation of the active compound, which ended with a lot of papers, but without knowing what was the chemical component that was responsible for the activity. They weren't able to isolate that because they were doing a bio-guided fractionation. So we decided to do this using chemometrics to try to know to try to identify the metabolite responsible for the activity, and then we managed to pinpoint these two main metabolites they seem to be the responsible metabolites for the anti-leishman or leishmanicidal activity. And then once we managed to isolate these components, we still don't know what is the molecular structure. However, we managed to isolate these two, and when we did the bioassay of those two peaks that we isolated, we found that there were very potent compounds. We were talking about the 0.075 micrograms per milliliters. That was the amount that was needed to kill most of the parasites. So this was a kind of the culmination of what we had been searching for, because those were compounds that at the same time were in a small concentration in the extracts. They were in a very small concentration, and we managed, using chemometric, to pinpoint the active ingredients. We did the isolation. It just happened that they were the active compounds, and this we already published it, and so this proves that we can use chemometrics and metabolomics as a new tool or an alternative to the bio-guided isolation, because it's quicker, easier, and it will save you a lot of time, and at the same time you will generate less chemical waste material that you will do otherwise using the traditional methods. However, again, currently there is a person trying to isolate more of these two compounds and to identify the chemical structure and to know. But again, what was really relevant is that they were not chemical components that were the major components in the plant extract. So we could use that as a way of even isolating or identifying compounds that are active, but they are very minor components in a full complex extract. So well, here in this picture there is most of the people that work with us. We have Dr. Maribel Vasquez, that she's the coordinator of the SARA. The Dr. Garcia Rodriguez, who will follow soon, Dr. Garcia Morales, and then we have Araceli, who is currently doing a PhD, and was mentioned before. We have Guadalupe Lupita, that she's the one running all the NMR experiments, and Claudia, Vero, and Edna, that they are normally helping a lot in all the functioning of the labs, because otherwise they wouldn't function. And we have Dorita, that helps us with all the admin work at at the unit. So, and again, and I would like to thank you for your presence and for listening. Thank you.
Gracias, doctor. Tenemos solo tiempo para una pregunta. ¿Sí? Bueno. Entonces, permíteme. Va a pasar las 3 conferencias y después todas las preguntas al mismo.
Okay, we will have the next conference, and then after that we will have the Q&A session with the two ponents.